Okay, have you ever actually stopped to think about the sheer unimaginable scale of the Earth's biological engine? Seriously, every single day, all around us, countless billions of plants are quietly pulling carbon straight out of the air and turning it into the very foundation of life on our planet. It's happening right now, literally outside your window. But here's the really fascinating part. As much as we talk about photosynthesis in these broad general strokes, scientists have figured out how to measure this completely invisible process with absolutely breathtaking precision. Welcome to This Explainer. Today, we're taking a journey into the mechanics of exactly how we decode this blueprint of breath.
So this is the core question that drives everything we're about to look at today. How do we actually measure plant life? I want you to put on your data detective hat for a second. We aren't just looking at green leaves and guessing if they're healthy. We're looking for the exact quantifiable mechanics of how scientists track carbon capture. How in the world do we put a hard number on something as wildly complex as an entire forest breathing?
Okay, let's dive into this. We're going to be looking at 1. The scale of photosynthesis, 2. Leaf-level gas exchange, 3. Measuring chlorophyll fluorescence, 4. Scaling up to ecosystem flux, and finally, 5. Open access data repositories. Let's start with section 1, the scale of photosynthesis, from the molecular level all the way to the ecosystem. This is our spatial anchor right here. Photosynthesis data doesn't just exist at one single level. It literally spans the whole spectrum, from the supermicroscopic molecular dynamics of harvesting just a single photon of light, all the way up to the carbon flux of a massive ecosystem. And because that scale is just so vast, researchers absolutely have to have structured measurement tiers for every single level of this biological engine. Moving on to section 2, leaf-level gas exchange. To actually capture this data, researchers use infrared gas analyzers, or IRGAs, that clamp directly onto a leaf. And we're not just looking at what a leaf is doing right at this exact moment. We want to know its full capacity. That brings us to a heavy hitter in plant physiology, the Farquhar-von Kammerer-Berry, or FVCB model. I know, it's a mouthful. But this mathematical standard derives Vcmax, which is the maximum rate of Rubisco carboxylation, and Jmax, the maximum electron transport rate. Basically, it tells us the absolute upper limit, the top speed of that plant's engine. But what everyday metrics actually feed that model? Well, think of it kind of like taking a plant's breathing rate. There are 3 core metrics we care about. First is A, the net CO2 assimilation rate, and we measure that in micromoles. Then there's GS, or stomatal conductance, which measures the gateway for gas exchange in moles of water. And finally, CI, the intercellular CO2 concentration, measured in parts per million. So you've got the inhale, the gateway, and the internal capacity. All right, section 3, measuring chlorophyll fluorescence and PS2 photochemistry. Now, what's really interesting here is this highly specific number, 0.83. We're zooming straight into the plant cells now to look at photosystem 2, using something called PAM fluorometry. Scientists use this to measure the maximum quantum efficiency of photosystem 2, which is represented as Fe over Fm. If you measure a perfectly healthy, completely unstressed leaf in the dark, you will almost always hit this exact baseline of 0.83. It's essentially a universal constant for plant health. So, what happens when a plant is out baking in the midday sun? Well, that brings us to its energy and stress dynamics. We're looking at 2 main things here. First is the effective quantum yield, or phi psi. This represents the fraction of ambient light the plant absorbs that is actually being used in photochemistry, so how much light is being put to productive work. But what if the sun is glaring down and it's simply too bright? That triggers NPQ, or non-photochemical quenching. This is a genuinely fascinating natural defense mechanism where the plant safely dissipates all that excess light energy as heat. Think of NPQ as a built-in pressure valve. It keeps the plant from getting sunburned or suffering severe photodamage. Honestly, it's an incredibly elegant survival tool. Section 4, scaling to ecosystem flux using eddy covariance. Okay, we're drastically zooming out now. We're moving from a tiny little clamp on a single leaf to massive eddy covariance towers monitoring entire landscapes. And at this massive scale, we have to contrast 2 really vital metrics. GPP, or gross primary productivity, is the total carbon fixed over time. Think of it as the ecosystem's gross income. But as we know, plants also respire. So we have NEE, or net ecosystem exchange, which is the net balance between uptake and respiration. NEE is the actual net profit, telling us for sure if that forest stored more carbon than it released. And finally, section 5, open access data repositories. We are literally collecting a mountain of data across all these tiers, and this matrix shows exactly where you data detectives can find it. It perfectly matches the zoom levels we just walked through. For leaf-level traits like VCMax, you go to the Tri-Plant Trait Database. For those massive ecosystem net profits we just talked about, you'd access FlexNet or Ameriflux. For canopy reflectance, you explore Ecosys. And to zoom all the way down to molecular transcriptomics, you dive into NCBI Geo or PRIDE. The coolest part, it's all openly accessible. Which leaves us with this final, really vital thought. We actually have the technology right now to map carbon processing from the efficiency of a single microscopic cell all the way up to the breathing rhythm of the entire globe. The sheer volume of open source data being shared every single day is staggering. But the big question we have to ask ourselves now is this. As climate challenges continue to grow, do these intricately measured data sets hold the ultimate key to understanding and maybe even securing our planet's biological future? The blueprint is there. It's just waiting for us to read it. Thank you so much for joining me on this explainer. I really hope you found the mechanics of photosynthesis as fascinating as I do. Keep exploring.